Hi everyone, today we're going to be talking about translation. What we're going to be doing is covering pages 78 through 80 in your lecture course paths. So if you look at page 78, at the bottom of that page, there is the start of translation and taking a closer look. The reading for this is concept 17.4. Before we take that closer look, let's just quickly review what translation is. So translation, right? In transcription, we made RNA from DNA. And now in translation, that RNA is going to be used as a template to make protein. So in general, here's what translation is. If we decode what that means and put it into words, we would say that we're getting protein synthesis from an RNA template. And the major player is going to be the ribosomes. So the ribosomes will be the major player. One of the major players. We'll also have the three types of RNA involved. And just to review ribosomes, right? We learned about these in Unit 1. Ribosomes are made of RNA and protein, and their job is protein synthesis. In prokaryotes, ribosomes are 70S in size. So prokaryotes have 70S ribosomes, but in eukaryotes, they have 80S ribosomes. That just means that the subunits of the ribosomes are a little bit different size. They have slightly different structures. The reason that this is relevant is because antibiotics that target prokaryotic ribosomes, like the streptomycin that you used in your lab, streptomycin will inhibit bacterial ribosomes, but it'll leave eukaryotic ribosomes alone. And this is important because when we take an antibiotic, we want to make sure that that drug isn't harming our own cells. All right, so let's draw a picture of what these ribosomes are, and then we'll get into that closer look of how translation occurs. So ribosomes, they are made of two subunits. We've got a large subunit, and then we have a smaller subunit. Both of these subunits, so we've got the large and small subunits, are each made of RNA and protein. We've got ribosomal RNA as the RNA in the ribosome, and then protein is another structural component of the ribosome. So here's one of our types of RNA right here. It's an actual part of the ribosome. All right, and then I'm going to change colors here. We've got, use pink, right? The messenger RNA kind of goes in between the large and the small subunits, and the messenger RNA is read from the 5' to 3' direction. So that's our messenger RNA. And go back to purple. In the ribosome itself, we've got three particular sites. From right to left, it spells ape. So first we have our A site. We'll define these later. Then we have our P site. And then we also have the E site. All right. In your book, I'm going to show you back right here. In your book, the A site is referred to as the amino-acyl tRNA binding site. I'm going to put that into words that make a little more sense. The P site refers to the peptidyl tRNA binding site, and then the E site stands for the exit site. So that one's pretty self-explanatory. So the messenger RNA binds in between the large and the small subunits. All right, so here's our picture. We've got the ribosomal RNA that's in the ribosome, and we have the messenger RNA, which carries the codons for protein synthesis. And so one more thing I'll add is that on our messenger RNA, that's where we have our 3-letter codons that are going to get translated. And so each one of these is read from left to right. So each of these right here are going to be the codons on the messenger RNA. So the tRNA is the one RNA we haven't fully discussed yet. We did a little bit of an overview of tRNA in lab. Here's the slide on transfer RNA in your PowerPoint. Transfer RNA is what I call the translator molecule. So you have a 3-letter anticodon that recognizes the messenger RNA codon. On the other side of that of that transfer RNA, you have where the amino acid is attached. So the anticodon is a triplet of nucleotides that is complementary to the messenger RNA codon. And the other end is what carries the amino acid or has that amino acid attached. Codon recognition, so now we are on page 79 in our course packs. Codon recognition is going to refer to the recognition of the messenger RNA codon by this anticodon. So hydrogen bonds will form or base pair between the anticodon and the codon. All right, so we'll draw that out here. I like to draw my tRNA molecules as stick figures. And so that tRNA that was just on the previous slide, right? I like to draw them as like a 3-legged stick figure with no arms. This is not an actual accurate diagram of what a tRNA looks like. But at the one end of the tRNA, here we go, we've got our amino acid that's attached. Right? And then at this end, we've got the anticodon. All right, so the one on the previous page was AAG, was the tRNA that was just shown. And anticodons go from the 3' to 5' direction because they're reading the messenger RNA codons that are 5' to 3'. So just like in DNA base pairing, they're going to be anti-parallel. So here's the tRNA anticodon. If we follow the rules of base pairing, right? So here's our messenger RNA strand that's going from 5' to 3'. That means that the codon will be UC, right? And these are hydrogen bonds forming that recognition process. Okay, so if you look at your genetic code, so I'm going to go back to slide number 5. Here's your genetic code. UUC right here codes for the amino acid phenylalanine, or PHE for short. So that means this tRNA molecule right here with this particular anticodon is going to be carrying the amino acid phenylalanine, and it's going to bring that to the ribosome. Okay, so the anticodon is the part of the tRNA that recognizes the messenger RNA codon, and then codon recognition is this process that's happening right here. So codon recognition is that process right there. The recognition of a messenger RNA codon by the tRNA anticodon. Okay, so here's an overview of what's happening as a whole. So we've got how one amino acid is brought to the ribosome in this picture right here through codon recognition. Here's an overview, and then we're going to go step by step. Here's a ribosome with the A, the P, and the E site. The existing polypeptide chain is in the P site. So P often stands for the existing polypeptide chain. All right, so that is resting in the P site. In the A site is where we have a tRNA molecule coming in, recognizing that codon right here, and bringing the appropriate amino acid. So this codon recognition process we referred to happens in the A site. And then a peptide bond is going to form between the polypeptide chain and the new amino acid that's brought in. Because peptide bond formation is involved in anabolism or the building of a protein, this is going to involve the use of energy because anabolism requires energy. We're not using ATP, but instead we're using GTP. But like ATP, GTP is a nucleotide triphosphate that has a lot of energy to offer. So it's another energy resource for the cell. All right, so when the peptide bond forms, now the polypeptide chain is in the A site. So what we're going to need to do is shift these tRNAs over. Actually, what happens is the ribosome moves over by one set of three nucleotides. So the translocation process also involves GTP, and when translocation occurs, the tRNA that was in the A site moves over to the P site. The amino acid or tRNA that was in the P site moves over to the E site. This tRNA has no more amino acid on it. So it's called an empty amino acid, and in the E site, that tRNA moves over. All right, so here it is one by one. In the A site, we have the process of codon recognition occurring. So that is happening in the A site. All right, and then the next thing that occurs, that peptide bond formation between the new amino acid and the existing polypeptide chain right here. And this is going to involve the use of energy in the form of GTP. And then finally, what happens is translocation. And in translocation, the tRNA from the A site is moved over to the P site. The tRNA in the P site is moved over to the E site. And that tRNA in the E site is then exited or checked from the ribosome like this. All right. So that is how an individual amino acid is added to a growing polypeptide during translation. All right, and so here's our lecture course pack again. If we put some of this into words, just to summarize, in our A site, this is going to be the site of codon recognition. And it's when a new amino acid, it's the site where a new amino acid is brought to the ribosome during translation. All right, so the letter A is for amino acid, and this is the one that's, if we just draw this here, here's our large subunit. We have the A site, the P site, and the E site from right to left. Okay, and then in our P site, this is the site where the existing polypeptide chain exists. Before peptide bond formation. Right? After peptide bond formation, that polypeptide chain moves over to the tRNA that's in the A site. All right, and then the E site is when after translocation. So in translocation, what we have is the tRNA that's in the A site moves over to the P site. The tRNA that's in the P site moves over to the E site. And in the E site, this is where that empty tRNA exits the ribosome. And then we can just start over by adding a new tRNA in the A site, and codon recognition will occur on the next messenger RNA codon. All right, so P site, I like to think of P as where the existing polypeptide chain is before peptide bond formation, and the E site is where the empty tRNA exits the ribosome. So to summarize everything that we've learned in both transcription and translation, in a eukaryotic cell, transcription is going to occur in the nucleus. It starts at the promoter region and it's going to end at the termination sequence. All right, in a prokaryote, this is going to happen in the cytoplasm because prokaryotes don't have a nucleus. We have RNA polymerase is the major player in this process, and RNA polymerase is going to make that messenger RNA or that RNA transcript. In a eukaryotic cell, where the transcript has to be shipped out into the cytoplasm, we're going to have some RNA processing steps or post-transcriptional processing steps, modification steps. And in these steps, the exons are spliced together and the introns are removed. So the final transcript with its added poly A tail and 5' cap goes through the nuclear pore and into the cytoplasm. At that point, a ribosome clamps on and translation occurs through this codon recognition process, followed by peptide bond formation, followed by translocation. So if we go back to this slide right here, I'd like to summarize that. The major processes that happen during translation occur in this order. The first thing that happens is codon recognition in that A site. The second thing that happens is we've got the peptide bond formation between the polypeptide chain that's in the P site and the new amino acid in the A site. And then finally, we have translocation, where all of the tRNAs shift over, or the ribosome shifts in the other direction, and the empty tRNA exits the ribosome. Okay, and then we get a polypeptide chain. Translation starts at the start codon, right? So the start codon isn't going to be at the very beginning of a messenger RNA strand. It'll be maybe 50, maybe 100 base pairs in. But translation is going to start at the start codon, and it's going to keep going until one of the 3 stop codons is reached. When a stop codon is reached, an empty tRNA comes in, and since it does not have an amino acid, the polypeptide chain is just simply finished being formed, and the ribosome falls off the messenger RNA strand. All right, so finally, in the last part of this lecture, we've got different types of mutations and how they affect protein structure. The reading for this is concept 17.5. All right, so we've got different types of mutations. These mutations occur in DNA. So mutations happen in our DNA, but that will affect the messenger RNA that's formed, and then that might affect the protein or the polypeptide that's formed as well. So the first type of mutation is called a base pair substitution. In your notes, this might also be called a point mutation, or in your reading. All right, so it happens at a single point where a single nucleotide in a gene is altered and replaced by a different nucleotide. If this ends up coding for the same amino acid, it's called a silent mutation. So a silent mutation has no effect on the amino acid that's coded for, and therefore no effect on the polypeptide chain. So it's silent, there's no effect. All right, a missense mutation is when that single nucleotide chain results in a single change in the amino acid. And then a nonsense mutation is when that nucleotide chain results in a premature stop codon instead of coding for an amino acid. Okay, so I'm going to look ahead and show you some of these right here, and we'll talk about their effects. Obviously, if it's a silent mutation, then and the amino acid is the exact same, then that'll literally have no effect on the protein structure or function. So there's going to be no effect on the protein in the case of assignment mutation. So here's what we're comparing it to right here. If the DNA codon is TTC and it's mutated to TTT, the messenger RNA is one nucleotide different, but the amino acid that's coded for is exactly the same. This is lysine right here. In a nonsense mutation, we get the formation of an early stop codon, and this is going to be a pretty severe effect because you're going to get only a part of a protein made. And then the missense mutations can either have a little bit of an effect or it can have a major effect, like what you saw in your supplemental homework assignment where you did the mutation exercise on the sickle cell red blood cells or hemoglobin. So in a missense mutation, if you get a change from lysine to arginine, you'll notice that both of these are cationic. So this would have a pretty minimal effect if the chemical structure has similar properties. But if you go from lysine, there's a plus sign here, so this is a cationic or ionic amino acid, and then you go to a relatively polar amino acid, this could, or a basic to a polar amino acid, this might have a more significant effect because the chemical structure is much more different. It would be an even greater effect if we went to a nonpolar amino acid. Okay, so those are the different types of base pair substitutions, or as I said, point mutations is a synonym for base pair substitution. And then the last are frame shift mutations. So in an insertion event, nucleotides are added to a gene. It can be one or more nucleotides, but if it's one or two nucleotides, that'll change the complete set of nucleotides that we consider codons. And so every single amino acid after the insertion event will likely be different. Same thing with a deletion mutation. In a deletion, nucleotides are removed from the DNA sequence. If it's only one or two nucleotides that are removed, then that will change the entire set of codons from the point of the deletion. And so when your codons are changing because the set of codons are shifting over, then we call that a frame shift mutation. So both insertions and deletions are called frame shift mutations, and both are going to have pretty major effects on the protein that's formed because all of the amino acids that are coded for after the insertion or deletion event are changed. All right, so that is all I have for you for the translation lecture. In terms of your Lecture 3 review guide, make sure that you know how the process of translation is initiated, carried out, and terminated. You should know what the process of codon recognition means and how translocation occurs. And you should also be able to recognize different types of mutations in DNA that would affect the various protein sequences that would form from different types of mutations. And your lab exercise that you did before spring break, as well as that supplemental bonus assignment, will help you to identify different types of mutations. That's all I have for today. I hope you all have a really good weekend, and I will see you in our review session on Monday. Bye.